Hello, I am Mathieu Piel and I have a cut on my hand. The immune system has to fight microbes that might enter my skin at that place. To do so, dendritic cells, let's call them DCs, must eat the microbes and move to the closest lymph node all the way up my arm. What do they do to get there? Let's first look at this nice cartoon made by my friend Renaud, a scientist cartoonist, and I will let my colleague Anna Maria describe it.
DCs are sentinels of the immune system. They are very motile. They explore tissues and they also constantly eat by a process called macropinocytosis. This is how it looks like when observing red DCs in a mouse ear skin explant with blue lymphatic vessels. At that scale, they look like little dots that jiggle around. Let's look more closely at them. This drawing shows the DC surrounding matrix, essentially composed of collagen fibers. There are also other cells around, like fibroblasts. Overall, the space is very crowded, but DCs can move incredibly fast in this crowded environment. Look how they move through this collagen gel. They are very deformable. With their speed of 5 to 10 microns per minute, they are amongst the fastest cells in the body.
In fact, if taken out of the crowded tissue and placed on a petri dish, DCs do not move so much anymore. But if you place a roof on the head, they recover the speed they had in tissues. This is because they have weak adhesion and they need confinement to move. This was called chimneying. Our lab found that any cell, if prevented from forming focal adhesions and placed under confinement, can move as fast as DCs.
Look at these hair cells on adhesive substrate. All its mousy motors are recruited on stress fibers, and they produce strong contract forces that are not very useful to move. The same cells, if prevented from adhering and confined, can move as fast as DCs.
Now, myosin II flows, just like it does in DCs.
Because cells seem to thrive under confinement, we now spend a lot of time experimentally squeezing cells and studying their behavior. We found that when a cell is compressed between two surfaces beyond a certain deformation threshold, it starts to blab indefinitely, but it doesn't die. The confinement changes the cell's contractile state.
There is a major drawback to crowded environments. The cell nucleus also has to strongly deform. How do they do that? The drawing does not tell.
So, we build microchannels with constrictions to watch cells move through small holes. And we found that they assemble a strong actin structure around the nucleus when it deforms.
So it doesn't seem so hard to squeeze the nucleus after all. And cells survive this exercise very well.
But we found that there is a price to pay for that. And that cost is that during this process, the nuclear envelope blebs and ruptures, opening this barrier between the chromatin and cytoplasm, also leading to DNA damage. If cells are not able to repair both the envelope and the damaged DNA, they die from this process of nuclear squeezing, just like this one.
Then, one day, after peaceful life running and squeezing around, the dendritic cell eats a microbe. Look at this beautiful mass it has. It is called micropinosomes. This encounter with the microbe induces a dramatic change in the way the cell moves. No more micropinosomes. All the actin and myosin are now concentrated at the cell back, and this new organization drives the fast and persistent motion towards a single target, the closest lymphatic vessel. What happens after is for another story, and we do not know much about it. We know that dendritic cells find their way through the maze of peripheral lymphatic vessels and eventually reach the lymph node, where they finally activate the adaptive immune response.
A few weeks later, my wound is completely gone. Thanks to DCs, I didn't get any infection.